is everything.
Okay, we'll go back to basics. Just waiting for a minute. Let's see if somebody joins in within a minute. Then I think we should start. So any assignment you people are solving, papers you did not get time to read, that's fine. How long are you going to have these surprise tests?
There will be 4 surprise tests for each subject. So
Within a semester?
within a semester.
That's too much.
Sir, the surprise test
Whatever.
will be set for the semester. We cannot say when we'll have the surprise.
Yes, sir.
Of course, that's why it's surprise.
No, that mid-sem system was better. For at least we would know that yes, there is an exam now. We will study. Okay, fine.
I have to study all the time.
That is the thing.
Good enough, good enough. Let's see. So, coming to the basics of things, let's talk about PCR. So, why is PCR done?
To amplify a particular gene of interest or
That is
DNA
correct.
fragment of interest.
That is absolutely correct. So, to amplify a gene of interest, we perform PCR, polymerase chain reaction. So, if I want to plot this PCR as a graph, this is, let's say, the temperature, and this is the reaction progression, whatever. How would it look like? Initially, what would we have? Anybody?
The temperature would increase during denaturation.
Temperature. So let's
So
say increasing at
it in,
denaturation. So we will have an denaturation phase, which will be an initial denaturation phase, right? Around
yes,
95 degrees Celsius for let's say 5 minutes. Then what do we have? Really sorry for this bad drawing. Anyway, then we will have the denaturation phase. Then we will have what next?
the constant temperature annealing.
Annealing phase. Fine. Then we will have
uh, Extension like
yes,
addition of
extension phase.
primers.
Extension phase, and then there will be a final extension. And this will be repeated like 30 times, 40 times, depending upon how much copy number you have initially. So is that clear to all of you? Is that, I mean,
Yes, sir.
okay. So how is the annealing temperature decided? Anybody knows?
By measuring the percentage of GC content.
That is correct.
Like
GC content of what?
GC content of that like the DNA of fragment of interest.
Actually, the
Its composition.
GC content of primers. So, okay, so what will the GC content give us?
The melting temperature.
Melting
That is
temperature.
correct. That is correct. So how is annealing temperature related to melting temperature?
This we don't know. I have.
Annealing temperature is usually 5 degrees less than the melting temperature. You'll be reading this if you have not read it yet. So around 5 degrees less than the melting temperature is the annealing temperature of what you give in your PCR reaction, although these are subject to optimizations. What if you lower the annealing temperature? What would happen? Anybody knows? What if you lower the annealing temperature below this? What if you increase the annealing temperature above PM minus 5? What are you supposed to observe? Let's say, what if you increase the melting temperature? Melting temperature is now greater than sorry, the annealing temperature is now greater than the melting temperature, what would happen?
Then the
So it won't
primers will dissociate from that
denaturing.
DNA, from the complementary DNA.
Ingit, anything you have to say?
Sir, it doesn't make sense because melting temperature is the temperature at which it denatures, and annealing is the temperature at which it renatures. So
Right, that is correct. So the primers, they will not bind to the DNA, right? Am I correct in saying that?
Yes, sir.
Clear? So if the annealing temperature is higher than the melting temperature, even equal to melting temperature, so the primers, they will no longer bind to the DNA and the PCR reaction will not happen. You will not get any amplicons. Clear on that?
Yes, sir.
Well, then what will happen if we decrease the annealing temperature much lower than the melting temperature?
The whole double-stranded DNA will that will get renatured, for which the primer cannot bind to the complementary region.
Primer cannot bind. If the
Wait,
annealing temperature is very low than the melting temperature, then there will be something what we call as non-specific binding. So we'll have multiple amplicons, undesired amplicons, which we do not want. So that is also a problem. So is that clear to everybody? Please raise your hands if it is.
yes, sir.
Raise your hands if it is clear to everybody. It's just helpful for me to see if you're following. Yeah. 3 of you. What about the rest? Shinjan, are you here? Shinjan, Devnarayan. Can you guys hear me? Am I audible?
Yes, sir.
Yes, sir.
Okay, so I'd assume it's clear to everybody. I think Shinjan is having some problem with his mic. Anyway, so after you get your amplicon, you will run an agarose gel electrophoresis, right? Clear on that?
Yes, sir.
What will be the problem? What is the problem that you see if you have one copy of DNA? Can you see any band here?
No, sir.
No. 10 copies very faintly visible. You cannot make out. Then you make 100 copies, something is visible. 1000 copies, it's clear. Then 10,000 copies looks the same. So how would you distinguish between whether your you have 10 to the power 4 copies or 10 to the power 3 copies? How would you distinguish that? It looks almost the same, right?
Yes, sir.
Any idea how would you do that? Think about it.
We might check for the band width. Like if the band is a bit thick, then it is 10 to the power 4, and if it is a bit like
Can you
thin,
really make
or
that out in between these two bands?
actually that's not possible. Wait.
You have done things similar to this in your microbiology lab. You have studied it in your microbiology course.
We will extract the we will cut the gel slice, and then after like dissolving the gel and isolating the DNA, we might check for the absorbance.
Sir,
Take the OD.
sir,
बताओ.
sir, are we talking about the application of protoplast fusion, genetic engineering method, or mutation over here in the if we have to refer to microbiology.
Really, not really. What if you have a lot of colonies in a agar plate? What do you do?
Sir, can you repeat the question?
You know what is an agar plate, LBA agar,
Yes,
urea
sir.
botany agar? You know that? You know how to streak colonies or how to
Yes, sir.
spread? You know that? So let's say if you have got a lot of colonies on your agar plate, what would you do?
Means we can just like isolate those colonies and perform
Isolation is not possible.
no.
It's so many
আচ্ছা
colonies.
কোয়েশ্চেনটা কি?
অ্যাগার প্লেটে তুমি হচ্ছে স্ট্রিক করেছো বা লেটস সে স্প্রেড করেছো, একটা তুমি ইনোকুলাম নিয়েছো, ওখান থেকে 1% লেটস সে ইউ হ্যাভ টেকেন and you have then spread it in an agar plate. So you see a lot of colonies. So you see lawn formation, মানে ইন্ডিভিজুয়াল কলোনি নেই। তো কলোনি পেতে হলে কি করতে হবে? Yes, Pragya?
So, like if we add something which is, resistant to certain present inside the agar, and the rest would.
You could do that. You're right. You're right. We could do that. But what would happen? The ones which are surviving are resistant strains, isn't it?
Yes, yes, yes,
So they are not the same bacteria you started with.
yes.
They are resistant bacteria. Anything else? It's very simple. If I tell you, you will be like, we have studied about this.
স্যার একটা পার্ট কেটে নিয়ে ওটাকে আবার গ্রো করানো।
Subculturing শুনেছো? Serial dilution?
না।
Serial dilution জানো না?
Subculturing শুনেছি।
Serial dilution?
So we can run the agarose gel and the distance moved will be less for the band, like 10,000 bands.
10,000 bands, run the gel. Well, I'm not sure, but anyway, have you heard about serial dilution?
No, sir.
So this term we have not.
You have never heard about it. Okay, so let's see. I was not aware of this. Usually it is taught in microbiology. This is a test tube. Let's say it has 10,000 microbes. You take 1 ml of this and add 99 ml media. What will, how much will be the, yes, Pragya?
Sir, actually, today's lab, NKJ's lab only, sir was doing transformation. So, okhane sir, actually, amader just today only itself, we did till the step of this spreading thing on the agar plate, and after like tomorrow we will see the growth of the colonies. At apore sir amader একটা calculation দেখিয়েছে, TE calculation. There it comes the feature of cell number divided number of cell transformed divided by microgram of DNA. So okhane sir mentioned a portion that how to count, and this subculture this point was first mentioned in today's lab classes. So
There is something called dilution factor, right, in the calculation of transformation efficiency.
Yes, sir.
Yes,
Sort of.
yes,
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 yes.
yes. So today one group only did this lab, so the rest don't know actually technically. So.
I think, I mean, this should be known in microbiology. I'm not sure why you're learning it right now, but anyway, that's fine. That's not the point here. So if you take 1%, so like 1 ml in 99 ml of media, 1 ml of culture in 99 ml of media is 1%, right?
Yes, sir.
So how much bacteria now you have finally in the final tube? 10,000 initially ছিল, finally কত থাকবে?
1,000.
1% of 10,000 is 1,000.
100. Sorry.
100 টাকে যদি
এটা
এখন
করেছি, বাট নাম জানতাম না, নামটা বলেনি।
হ্যাঁ
ডাইলেশন ফ্যাক্টরটা দিয়ে করা এরকম করেছি,
ডাইলেশন ফ্যাক্টর দিয়ে মাল্টিপ্লাই করা, হ্যাঁ,
কিন্তু নাম বলেনি কখনো।
This process is called serial dilution. So now if I dilute, let's say 10 to the power 4 number of, I mean, this is the DNA, the copies of DNA, and I take, let's say, 1% of it. So how many DNA would I have? 10? Or no, sorry, 100? 100?
100.
Okay. If I do the same dilution here, I take 1% and
10.
then it will be 10. So 10 and 100 are distinguishable.
Yes, sir.
Yes,
So
sir.
see, that's why it's semi-quantitative. It's not total, it's not a quantitative process. It's semi-quantitative. Clear on that?
Yes,
Is
sir.
it okay? Everybody's following?
Yes, sir, but still we won't know the exact number.
Why would we not know?
I mean, we are comparing, suppose we have 10,000, we are taking 1% of it, and we
So
are
let's say we
taking
have 10
1%.
to the power 6. We don't know how much we have, but we are taking 10 to the power 6. Or let's say 10 to the power, let's say 10 we are taking. Okay, that's a better number. 10 to the power 10. I took 1% of this. So that would be 10 to the power 8. 8 or 7. How much will it be? 8, I think.
8 into the
Yes,
0.
sir. 8.
Then I'm doing again 1% I'm taking of this 10 to the power 6. Again I'm taking 1%, 10 to the power 4. Again I'm taking 1%. Then it is coming as 10. So the number of serial dilutions I have done will let me know that what is the final number of copies.
Yes, sir.
Was asking this Ingit, right? You're clear on this?
Yes, sir.
Okay. So, but anyway, it's a tedious process. So 10 to the power 10 থেকে start করবো, 10 to the power 8 টাকেও জেলে রান করবো, 6 ও রান করবো, 4 ও করবো, because আমি জানি না initial কত ছিল. So it is a very tedious process. So anyway, quantitation is a difficult process in case of agarose gel electrophoresis. So what do we do? Any other kind of technique which will help us see, which will help us quantitate better?
Can
স্যার
you
পিসিআর এর কোনো উল্টো ডিভাইস আছে?
PCR এর
কোনো উল্টো ডিভাইস আছে এরকম কিছু যেটা কমায়? খুব বাজে কোয়েশ্চেন ছিল।
বাবারে।
if I answer this, you will know this. You এটা যদি শোনোনি বলো তাহলে আমি মানবই না।
Preparative, like low melting point agarose is for preparative gel.
Low melting point agarose is a separate concept. I mean, let's not get into it. কোভিড এ কি হয়, কি পিসিআর বলে ওটাকে? কোভিড টেস্টে
RT-PCR.
what is RT?
PCR.
Reverse transcription.
Reverse
No,
transcriptase.
RT is not reverse transcriptase. RT is real time.
For real time.
Oh, real time.
Yes. This is how a real-time PCR amplification curve from a real-time PCR looks like. So this is the threshold. I'll tell you why it is real-time, why it is not RT-PCR as such. I'll tell you why that is. So what do we see? This is a sample A and sample B. Let's say it's for COVID only. There are samples taken from two different patients, A and B. So which particular sample has a more amount of initial DNA or RNA? In this case, it would be RNA.
10 to the power 10 things.
10 to the A, sample A or sample B?
স্যার এটা মানে এই গ্রাফটা কি রিপ্রেজেন্ট করছে সেটা বুঝতে পারছি না, এটা একবার একটু বলবেন?
Okay.
Sample
Okay.
A.
This is the cycle number. That is the number of cycles you have gone through. So in this entire reaction, they went to around 45 cycles. It looks like that. It's around 45 cycles. Clear? Clear on that?
Yes, sir.
Now they have used a fluorescent dye which binds to the double-stranded DNA. Fine. So when more number of double-stranded DNA is formed, more fluorescence will be there. Clear on that?
Yes, sir.
So now there are two samples, sample A and sample B. We will talk about real-time chemistries very soon. Sample A and sample B. Now sample A, it crosses the threshold value at, let's say, 20th cycle or 22nd cycle, whereas sample B crosses the threshold value at 32nd cycle. So which has more number of DNA or RNA as a starting material, sample A or sample B?
Sample A.
Sample A. So is that clear with everybody? Because sample A, it has more number of initial copies of DNA or RNA. Hence, it is forming duplex structure earlier than sample B. Clear on that?
Yes, sir.
Yes, sir.
Then why do we have this threshold? This threshold is there because initially when you will measure the fluorescence, when DNA duplex have not happened, there is something called primer dimers which could form. Primer dimers might also give fluorescence. There could be other kind of dimerization processes that could happen, which will give you fluorescence. There will be a noise. So we decide a threshold value above which we will consider that amplification is taking place, below which we will say that amplification is not going on, something else might be happening. So have you seen CT values in a COVID RT-PCR report?
তো তার আগে একবার বলবো
Yes, sir.
নয়েজটা কি স্যার? জাস্ট লাইক এটা আমরা
Error.
বায়োইনফোতে শুনছি বাট ক্লিয়ার নেই কনসেপ্টটা।
Info the noise.
হ্যাঁ
না,
স্যার।
ওটা actually error basically.
Error
Error
না,
ছিল।
এররও হবে না। Noise is like primer, যখন primer dimers হতে পারে জানো তো?
হ্যাঁ।
Do you know the concept of primer dimers? You have a
Yes, sir.
forward primer, you have a reverse primer. Let's say if it is overlapping, তাহলে তো হওয়ার chances অনেক বেশি। Overlap না করলেও, since they have a similar melting, temperatures, so if the primers are not amplifying the DNA, what could happen? These primers themselves could form dimers. এবার প্রাইমার, ওই জন্য ফরওয়ার্ড প্রাইমার আর রিভার্স প্রাইমারে যদি অনেক বেশি কমপ্লিমেন্টারিটি থাকে, if it has a lot of complementarity, তাহলে ডিএনএ অ্যাম্পলিফাই হবে না, প্রাইমার ডাইমার হয়ে বসে থাকবে। ক্লিয়ার? মানে ইজ ইট আন্ডারস্ট্যান্ডেবল?
হ্যাঁ বাট তাহলে কি ওখানে হিট দিয়ে ওটাকে ছাড়িয়ে দেবো?
হিট দিয়ে ছাড়ালে দেখো ব্যাপারটা হচ্ছে, যদি কমপ্লিমেন্টারিটি বেশি থাকে হিট দিয়ে তো ছাড়িয়ে দেবে, কিন্তু ওটা কি ডিএনএ কে বাইন্ড করবে? করবে তো না।
না। দেন তাহলে বাড়ছে কিভাবে?
মানে স্যার হায়ার টেম্পারেচারে, মানে প্রাইমার টাইমার ফর্মেশনটা তো হায়ার টেম্পারেচারে হতে পারে
না, এরকম কোনো মানে নেই। টেম্পারেচার কম করলে, বিকজ হায়ার টেম্পারেচারে ডিন্যাচুরেশন হয়। So when you are going with the denaturation phase, everything gets denatured. So the phase is important for this reason only, so that you can free the primers. Primers gulo beriye ashlo, then they are free for binding to the DNA template. Are you understanding this?
মানে আমরা যখন like when we are adding primers to that like maybe test tube or something, whatever microtest tube, along with that specific gene of interest and all, at that point of time the primers may be complementary to each other, means since we do not know the sequence of the DNA fragment, so we are adding some random primers which might be complementary, for which primer time information occurs, right? But then how do we understand this threshold value?
Threshold value is usually when you run an RT-PCR, what happens is that you will see a lot of noise will come up. So primer dimer is one of the reasons. Another reason is that your initial template, the starting point of the DNA, it only had a lot of, it was also double stranded, isn't it? So that could also cause formation of a double, it would also give fluorescence, right?
Yes, sir.
That is one reason. Another thing, you are adding DNTPs also.
Yeah, yes, sir.
These DNTPs, what they are forming, what kind of dimers they are forming, we don't know. So there is noise in the lower level. So this happens in a lot of like electronics concept also. When you go below this threshold value, you really cannot make out that whether amplification is happening or all these kind of problems are happening. But if it goes beyond the threshold value, because these kind of problems are minimal, threshold value upore chole gale, then we know that amplification is happening. The fluorescence is due to amplification only, not due to other reasons that we have stated. Clear on that?
Okay, sir.
So have you seen a CT value in a COVID report, COVID RT-PCR report?
Yes, sir. CT value देखी थी, but
What does it signify? Have you ever tried to
it's
figure that out?
it signifies that threshold value, I guess.
Threshold cycle number. So this, let's say if it is 22 for sample A, and it is, let's say, 32 or 31 for sample B. So this 22 is the CT value for sample A, 31 is CT value for sample B. Clear?
Okay, sir.
Now, what would a CT value signify in your RT-PCR report? Think about it. Let me rephrase the question. If you have a CT value, like two people had COVID here, let's say person A and person B. So person A had a higher amount of COVID. I mean, he was in a higher state, in a more severe state, where COVID virus, it has replicated more, while person B is on a less severe stage where virus has not replicated as much as it could have. So you do an RT-PCR test. What would you expect? Whose CT value would be lower?
Person B.
Person B who had low COVID virus yield, or
So wait. Let me think.
যার সিটি ভ্যালু বেশি তার বেশি সিভিয়ারিটি, সো এ।
ঠিক উল্টোটা
কেন?
তো হ্যাঁ,
কেন যদি ভ্যালু বেশি হচ্ছে তো উল্টো কেন? লাইক সিটি
if you
ভ্যালুটা কি
have
আরেকবার?
more amount of, let's say, viral load in your blood or whatever in your nose swab, whatever they are taking as a sample, blood is not the case here, but in your nasal swab, if you have more number of viral particles, then what would happen? Amplification will take less time or more time?
লেস।
The CT value will be lower or higher?
ও লোয়ার।
Lower. Achha.
If the CT value is lower, that means the severity of your disease. So we can also predict that using the CT values. That is how RT-PCR is so beneficial. Even though if it is expensive, still RT-PCR is one of the gold standards for diagnostics.
তো সিটির ফুল ফর্মটা কি?
Threshold cycle. So it's the cycle number at which your sample has attained the threshold value. This point. See CTA, CTB. Can you see that? Clear on that?
Yes.
Okay, good enough. So what is the limitation with traditional PCR? Okay, another thing that I missed in the previous one. So agarose gel will give you only the result which happens at the end of everything has ended. Whatever you can see is what you see in an agarose gel, right? So agarose gel detection, a normal PCR will give you end point detection, right? Is everybody following me?
Sir, এটা কি বললেন বুঝতে পারলাম না।
If you go for a traditional PCR, not a RT-PCR, where you do not have this dye which can bind to the, this double-stranded DNA, and it cannot give fluorescence. So what would you do? You would run the the final product in an agarose gel.
Yes, sir.
So the final product you will only get to know in case of a normal PCR, in case of a traditional PCR, Like if you take the example of sample A and B, in the end, both of them have similar fluorescence, right?
Yes, sir.
So in the end, so agarose gel electrophoresis only gives you a endpoint detection. I mean, is that clear or is it not?
Yes, sir. Yes, sir. Yes.
While RT-PCR will give you a real time. RT-PCR is also known as qPCR. So it gives you a real time detection. So a traditional PCR, it will give you an endpoint detection. It is best for qualitative testing, whether you have the thing or not. That is fine. But how much you have the DNA, how many copies of DNA you have, that you cannot find out. So it's not ideal for quantitative analysis. So is that clear to everybody? Is it clear?
Yes, sir.
Okay, so a real-time PCR where we monitor amplification using fluorescence techniques. So we use fluorescence dyes for this purpose, and we quantify initial amount of template by monitoring the amplification curve. We have seen the amplification curve. So that's how we find out how much of DNA was present initially. So any dye which you know which is used for real-time PCR?
Ethereum bromide.
I think I have only talked about it.
Cyber green.
Cyber green.
Cyber green, it's a cheap, inexpensive dye. So anyway, this is the amplification curve. So we already talked about it. So which of these 1, 2, 3, 4, 5, which of these had the highest amount of DNA as a starting material? DNA or
One.
RNA? One.
One.
Which has the lowest?
5.
5.
5. Good enough. So what is a real-time PCR? It's nothing but a PCR with a camera. And of course, fluorescence dye is involved, nothing else. So it's usually a PCR, a normal PCR. If you add a camera to it for detection, and you have your fluorescent dyes or probes, we also use probes. So in those cases, we get an RT-PCR. This is how the setup looks like, right? So although it's very much more expensive. So if we can plot a standard curve, you know what standard curve is?
Yes, sir.
So if we can plot a standard curve between CT and the copy number, if we plot a standard curve, then we can find out for an unknown sample, if we get a particular CT value, what would be its the initial copy number? We can find it.
Yes, sir.
So that is understood by everybody, right?
Yes, sir.
So for real-time PCR chemistries, as I said, the dyes or probes which are used, one of the most used ones is cyber green. Fine. So as I said, it just binds to the double-stranded DNA. Whenever double-stranded DNA is formed, it just binds to it and it gives you fluorescence. If double-stranded DNA is not formed, then it will remain as free molecule and it will not give fluorescence. Clear?
Yes, sir.
What does a TACMAN probe do? TACMAN probes and molecular beacons, these are specifically designed. So we'll talk about it later. But let's talk about cyber green now. So it only fluoresces when it is bound to double-stranded DNA. It's really cheap. But what could be one of the disadvantages which could happen if we use cyber green?
DNA ভেঙে গেলে ওটা থাকবে না fluorescence.
DNA ভেঙে গেলে ঠিক আছে, আর কি হতে পারে? We talked something about annealing temperatures while ago, no?
Oh, yeah.
So what happens if the annealing temperature is lower?
Non-specific binding.
Non-specific bindings or non-specific amplification. So what would happen if you have cyber green in your mixture? Will it be able to distinguish between your target amplicon
No.
and the non-specific amplicon?
No, sir.
No.
Because
So it could give,
everything.
yeah, it could give false positive results. Clear?
Okay, sir.
Okay, good enough. So this is how a Tachman probe works. So a Tachman probe, it contains uh, a fluorescent molecule and a quencher molecule. And this Tachman probe also has, it is designed like a primer. For each reaction, it will only bind to your target DNA of interest. Clear? It will not bind anywhere. So what would happen when extension will be going on, when the extension phase will be going on, what would happen? Let's say if the polymerase is extending here, polymerase is extending the DNA, and then what would happen? This would break. Can you see the third one? Are you following me on this or not?
Yes, sir, yes, sir. They
Sir, can you
It will
repeat this
polymerization.
break.
from?
So in
Sir,
this case,
can you
yes,
can you repeat this from the beginning? I couldn't understand.
Tachman probe, it has a reactor, it has a fluorescent molecule, it has a quencher molecule. When these two are close together, there will be no fluorescence. Fine, because the quencher will not allow the fluorescent molecule R here to fluoresce. Fine. But when there is extension, what would happen? The polymerase would start polymerizing, DNA will be formed. Due to this fluorescent molecule, it will be free from the quencher. And once it is free from the quencher, now fluorescence will be there. Clear? কি এখনো বুঝতে অসুবিধা হচ্ছে?
ঠিক আছে, বুঝেছি।
So okay, so at what step do you think that we can check? Okay, we will come to it later, but the thing is, what could be the disadvantage of this process, Tachman probes?
যদি polymerization না হয়,
তাহলে তো পলিমারাইজেশন না হলে there will be no fluorescence, তো তার মানে তো ঠিকই আছে, ভুল তো কিছু নেই।
স্যার একটা জিনিস জাস্ট আমি বলুন যে যখন মানে ইনিশিয়ালি কোয়েঞ্চারের জন্য ফ্লোরেসেন্স হচ্ছে না, কিন্তু তারপর যখন প্রোবটা গিয়ে বসছে, তারপর আমার কোয়েঞ্চার মলিকিউলটা মুভ করছে না, আর মানে
এই যে
ফ্লোরেসেন্ট।
আর আর কিউ যেটা হচ্ছে তোমার টেমপ্লেটের সাথে বাইন্ড করা আছে না, তো যখন পলিমারাইজেশন হচ্ছে তখন কি হচ্ছে যে এখানে পলিমারেজ তো এখানে ডিএনএ অ্যাড করে দিচ্ছে, তখন কি হচ্ছে এই যে এটা ব্রেক হয়ে যায়, তো কিউ ইজ সেপারেট নাও, আর ইজ সেপারেট, তো আর এর ফ্লোরোসেন্সটা আসছে। তো যেটা সাইবার গ্রিন ফ্রি ভাবে ফ্লোরোসেন্স দেয় না, এখানে ঠিক উল্টোটা হয়। তো যদি আর ফ্রি থাকে, হ্যাঁ
ব্রেক কি করে হচ্ছে, মানে পলিমারাইজ তো করছে না, আমি গন্ডগোল করছি।
এখানে সি দিস ইজ দ্য ডিএনএ। So let's say if we have, it's very difficult to make you understand like this. So this is, let's say, the DNA, এর উপরে হচ্ছে বসে আছে একটা probe. This is the probe, ঠিক আছে, which is sitting over it. It is of a very small size. So let's say like this. So what is happening when the polymerization is happening? তখন because of that stress that is getting on, which is given on this probe breaks. It is not removed from it, but it rather it breaks. It is designed in such a way. Clear?
Okay, okay.
Okay. So what could be the disadvantage? সাইবার গ্রিনের অ্যাডভান্টেজ কি ছিল?
চিপ, এটা এক্সপেন্সিভ প্রসেস।
এক্স্যাক্টলি, তো এটা এক্সপেন্সিভ, এটাতে বেশি স্কিল লাগে, obviously you have to design a probe also. So it will be cyber green I can do for n number of RT-PCR reactions, but Tachman probe I have to make for individual reactions. So is that clear on that end?
Yes, sir.
Okay. What could be the other disadvantage of Tachman probe? রিউজ করা যাবে?
না, রিউজ করা যাবে না।
So see, this is another disadvantage. So it cannot be reused. Acha, one more difference I want to find out, at which stage would I read? So the fluorescence reading ta hobe na. You have seen this PCR. I have drawn this graph. This one, right? So this is the number of cycles that we have. Initial one is denaturation, then it's annealing, then it is extension. So cyber green e কোন stage আমি যদি reading নি তাহলে best reading আসবে?
Annealing stage. Sorry, extension stage. Sorry.
ঠিক আছে, এখানে cyber green এর reading নিতে হবে. Everybody is understanding this?
Yes.
What about Tachman probes then?
Annealing.
ওটা annealing এ।
কারণ জাস্ট ভেঙে যাবে যখন তখনই বেটার আসবে।
I think it would be denaturation, no?
কিন্তু denaturation
Extension
এ
পুরোটা হয়ে
polymerization
গেছে।
হচ্ছে।
এক্সটেনশনটা পুরোটা হয়ে গেছে। এক্সটেনশন পুরো যদি না হয়, সাইবার গ্রিনে এক্সটেনশন পুরো না হলেও তুমি একটা রিডিং পাবে which is giving you some amplification result, right? But এক্সটেনশন যদি পুরো না হয় তাহলে at the denaturation state যখন কি থাকবে, there will be the template, there will be some of the probes which will remain like this. So these probes which have initially not bound to the DNA at all, they will not fluoresce. But at the denaturation state, যেই probe গুলো bind করেছিল সেগুলো ভেঙে গেছে, আর তাদের যে R value, R যে molecule, fluorescent molecule টা free আছে, they will give fluorescence. Clear?
না স্যার, এটা আমি বুঝতে পারলাম না।
পুরো এক্সটেনশনটা যদি না হয়, see, this is the extension step, না? See, we are in the middle of the extension step here. You can see this.
Yes sir.
So if this entire, let's say you have designed a probe for this part. So at this stage, if you read, if at the stage of extension, if you take a reading, then what would happen? Fluorescence কি আসবে?
না স্যার, fluorescence আসবে না।
কিন্তু amplification তো হচ্ছে।
আচ্ছা, হ্যাঁ হ্যাঁ বুঝেছি।
তো আমি যদি এই স্টেপটার পর আবার যখন এখানে এই স্টেপে চলে আসবে ডিন্যাচুরেশনে, তখন কি হচ্ছে? যেই প্রোবগুলো ইউজ হয়নি সেই প্রোবগুলো এরকমই থাকবে, তাদের কোনো ফ্লোরোসেন্স নেই। বাট যেই প্রোবগুলো ইউজ আপ হয়ে গেছে তার আর ভ্যালুস গুলো, মানে আর ভ্যালু ইজ বেসিক্যালি দ্য ফ্লোরোসেন্ট মলিকিউল, ইট উইল বি সেপারেট, রাইট?
হ্যাঁ হ্যাঁ, ওকে।
So denaturation would be a better step. মানে is it clear or is it not?
Yes sir, yes sir, understood.
In the cyclic process, denaturation, we take the reading for Tachman probes. What would happen if we do the same for cyber green? Can we do the same for cyber green?
No,
No sir.
because cyber green তো double bond এর সাথে হবে।
Correct. So I hope everybody understood this. Or
Yes, sir.
is this okay? Now we have something called molecular beacons, very similar to that of very similar to that of, just let me look. Just a minute. Did I miss out something? Okay. Yeah, very similar to that of the Tachman probe. So here also we have a fluorescent reporter dye and a quencher dye. So when nothing has happened, it will remain in this conformation. Fine. And in the middle, in the loop region, it will have some PCR specific nucleotides. So when PCR is happening, what would happen? That there is, again, let's go for it. There is denaturation, then there is annealing, and then there is, let's say, what is the next step? Extension. So while denaturation has happened and annealing is going on, at that time, what would happen is this will open up and it will bind to the specific target, and it will give you fluorescence. Clear?
Yes, sir.
I have one
But
question.
yes,
So why don't we use etherium bromide instead of this?
ethidium bromide কি করে? It's an intercalating agent.
What are the correlation?
Fluorescence দেয় UV light, yes, you're right. But what is the other problem with ethidium bromide?
Carcinogenic ছাড়া আর কি?
Carcinogenic, so definitely it's a reason. What else?
Sir, it intercalates, so composition টা তো চেঞ্জ করে দিচ্ছে না ডিএনএ এর
That is one reason. That is one
So, but
reason is
এখানেও
true.
তো এটা ইন্টারক্যালেশন হচ্ছে না, লাইক এখানেও তো লাইক ডিএনএ মাঝখানে গিয়ে চলছে।
Intercalation টা কি?
So
What is
like binding in
intercalation?
between DNA, right?
In between DNA what?
Base
DNA
stack. DNA base
base
stacking. In base stacking.
base
Uh-huh.
stacking, right? Base
In
stacking.
DNA base stackings.
DNA base stacking. So when you see the DNA, let's say this is one base, this is one base, this is one base. These are three bases. Ethidium bromide will bind here in between. Whereas what the cyber green does, it binds to the double-stranded DNA. It's different from ethidium bromide. Another problem that one of you pointed out is ethidium bromide এর জন্য কি হয়, when they're in between base stacking, if you have a dye present there, so there will be a gap between two DNA bases.
Yes, sir.
So if there is a gap between two DNA bases, the polymerase might think that base is absent, so it will go for, I mean, the repair process might be, initiated. তোর মাঝখানে কোনো বেস মানে ঢোকানোর চেষ্টা করবে। Clear, Shinjan?
Yes, sir.
Okay, so far so good. So what is one advantage of molecular beacons? Can it be reused?
Yes, sir. It can be reused.
Can be reused. It does not break. It can be reused. Yes. Yes, Mayuk.
Since probes are recycled, so we can say that molecular beacons can be reused again and again.
Yes, it can be reused. I mean, I did not get your question. What are you trying to ask? Is there a question there?
No, sir. I'm just answering that since the probes are itself are getting recycled, so molecular beacons can be used again and again.
Yeah. So fluorescence is red at the annealing step. Clear on that? Why is it red on the annealing step?
Yes, sir.
Okay, good enough. So what would happen if we read this?
Sir,
Stackman
sir, one
probes
thing.
in
How
annealing
can
step?
how can we use the molecular beacons? Use the molecular beacons, like when DNA polymerase will like add DNT base, like will it not break due to the stress?
They will not break because they have this loop kind of structure. So whenever polymerase would come up, what would happen in the extension step? What would happen is this will just go, a base ta just bhenge jabe, or this will again form this kind of a structure.
Okay. Yes, sir.
So some restriction enzymes which are methylation sensitive, these are HHA1, HPA2. These are methylation sensitive restriction enzyme. So we will see what happens with these kind of restriction enzyme, and there are some methylation specific restriction enzymes like MCRBC. We'll talk about both of them. So when we are talking about restriction enzyme digestion with real-time PCR, we have done a real-time PCR. So before doing real-time PCR, even before PCR amplification has begun, if a base is methylated, it will not be digested by HPA2. So HPA2 is essentially its enzymatic activity. I mean, its cleavage recognition site is CCGG 5' to 3'. So if C is methylated, any of the C's are methylated, then this restriction enzyme will not work. Clear on that?
Yes, sir.
So if it is not methylated, then this restriction enzyme, if you give, it will break the DNA and there will be no PCR product formed. But if there is a methylation present, then this HPA2, it will not digest this DNA and there will be PCR amplification. Is it clear, or I think there must be some confusion here?
No, it's clear.
Okay, so why can't we amplify target DNA and then digest it with restriction enzyme and look at the digestion pattern? The question which has been asked here. What would happen if I start amplification from here?
Because we don't know whether the methylation is present or not. And if methylation is not present, then that will be a problem, I guess. Maybe.
Is methylation, does it happen in a PCR reaction? Does it happen?
No.
So what would happen? Your templates, if you amplify this this initial template, you will get DNA like this. Majority of your template or the final product will be like this. So if you then go for PCR amplification, you will not get any product because you're adding HPA2. Clear on that?
Okay, sir.
Everybody understood? Or is it still not clear? Okay, I'm a little bit tired today because I had a lot of TA duty today. Anyway, is it clear with everybody or should I explain it once again? Okay, let's move on. So restriction enzyme digestion for DNA methylation studies, what is the advantage? It's cost effective, no special instrumentation is required, only an enzyme is required. And you can analyze specific target regions, like let's say P53 promoters, these kind of specific regions you can target, you can see in using this method. The limitation is the analysis is based on the on the presence of a specific restriction site. So if let's say instead of CCGG, if you have CAGG, so will the restriction enzyme cut this site?
No, sir.
No, sir.
No. But will there be PCR amplification?
Yes, sir.
Okay, so you might infer that there is a methylation present here. One might infer this because we don't know whether it's CAGG or CCGG, right? Sequencing has not been done yet. So
yes,
that could be one reason. Another thing that could happen if there is some mutation at the recognition site, as I said, then in those cases, there will be false positive results. And if there are multiple restriction enzyme recognition site, then the interpretation is very complicated. It becomes very complicated because a lot of bands come up. Some are amplified, some are not amplified. So if there are many restriction enzyme restriction sites, then we cannot really understand that what is happening, which site is methylated, which site is not methylated. Clear?
yes, sir.
So some methylation specific restriction enzymes, they will only target or they will only cleave the restriction sites which has methylated cytosines. So the thing is the recognition site is very non-specific in this case. So only GA, then there will be a methylated cytosine, and then there are 40 to 3000 nucleotide somewhere in between it will cut, and then there will be another GA and a methylated cytosine. So one good thing is that it is highly sensitive. Even small amount of methylation can be detected, and it can be useful for global methylation profiling. Let's say you are checking the methylation status of, let's say, Drosophila genes. So if you're trying to do something like that, this kind of enzyme will be more helpful because it will be giving you somewhat an idea that where methylation is present. Clear? Clear on that?
Yes, sir.
But the limitation is that it is not useful to study specific targets. So since its cleavage is between 40 to 3000 nucleotides, so can we really study promoters in this case? Maybe there might be multiple promoters. You will never find out. In between 3000 nucleotides, there could be a lot of things. So it is very difficult in such a case to find out whether methylation specificity is helpful. And it cannot be combined with PCR. Why so? Why cannot be combined with PCR? I think you people are not really following this.
Maybe no, this cannot be combined because if it cuts at no, if it doesn't cut at specific targets, then we will not be able to amplify a particular gene of interest.
Yes, that is correct. That is one reason. Another thing is methylated DNA is getting cleaved, right?
Yes, sir.
So methylated DNA, if it gets cleaved, if you go in this part, if methylated is getting cleaved, let's say we interchange the position here. So methylated is getting cleaved and unmethylated ones are not getting cleaved. So unmethylated ones will have PCR amplification. Methylated ones will not have PCR amplification. Clear?
Yes, sir.
How would you know whether that non-PCR amplification is due to your problem in primers, problem in the PCR reaction, or it is due to the activity of the enzyme? Clear?
Okay.
So this is how an MCR-BC digestion looks like. Lane 1 has human DNA. Lane 2 has human DNA incubated with MCR-BC. Lane 3 has Drosophila DNA, and Lane 4 has Drosophila DNA incubated with MCR-BC. So what is the inference that you can draw from here?
Upon this MCR-BC digestion, like the PCR amplification is very less because of the fact that
PCR is not done here, only agarose gel electrophoresis.
Okay, okay, only agarose gel. So in this case, like a large number of small fragments are produced in case of human DNA.
That means
that means there are that means human DNA contains means the methylation status or methylation profile of human DNA is much more as compared to Drosophila. More number of methylation is possible.
Is that clear to everybody here?
Yes, sir.
Okay, I will stop right here because I have an interactive session planned today as well. And anyway, I'm not really feeling well to teach further. We will take this up in the next session, the rest of the things. So we'll talk about bisulfite conversion, how it is important to find out methylation specific PCR, how we can do that due to bisulfite conversion. We will take up this topic later. Today we have Mr. Shomin Bid. He is a 2021 graduate from Heritage, and he did his MTech from IIT ISM Dhanbad in pharmaceutical engineering. And he's currently an assistant manager in Zydus Life Sciences in the Baddi campus. In Himachal Pradesh. It's very close to Chandigarh. So, Shomin, sir, can you hear me? Shomin, are you here?
Yes.
Okay, so since some of you had asked that if there is anyone whom you would like to interact who has a pharmaceutical industry based background, we have him here. So if you have any questions, this is the right time to ask. I will just stop the recording for the sake of everybody and let's see.